Yeah, so as I mentioned to you, today I'm just, uh, before I go deep into this particular topic of molecular diagnostic, just want to review what we discussed last week. So to begin with, we talked about antibodies, um, and antibody is a is proteinaceous in nature in majority, and it is excreted or secreted by B cells. B cells are lymphocytes. So if you remember stem cells, when we talk about oligopotency, uh, I mentioned to you that lymphocytes could have two branches, um, one mainly T cells and the other B cells. T cells mainly produces cytokines and interleukins, but B cells, they produce antibody. It's quite a big molecule, and it's Y-shaped. And if you look at it just at the surface, superficial, there are four chains, out of which two are light, two are heavy, and heavy has extra length of this heavy chain, which is called as constant region. The groove of the V within the Y, this is a place where the foreign particle binds, and foreign particle that has capacity to generate immunity or immune response, they're called as antigens. So when antibody is produced, then they try to go and go and bind to the pathogen or bacteria, virus. So this has the two lighter ones, which here is pink or pinkish purple color. They are light chains, and they consist of variable region and constant region, variable constant region. This variability is important um, for detecting various kinds of pathogens, especially variants. So there could be one virus, but it could uh, change its structure because of genetic changes, variations. So these antibodies, they can. So you can ask me one very interesting question that mutations are also a type of variations, right? Changes in nucleotides. So why here changes in nucleotide? Why you don't call it mutation? Why do you call it variation? So that's a very interesting and thought-provoking question. That mutations generally are generally which can traditionally mutations are, how do you define it? It's defined change in nucleotide. That could be anything. And usually, traditionally mutations are taught for bad effects, but mutations could have good effect. Here, it is not mentioned as mutations, but it is mentioned as variations, because it shows flexibility in our immune system, right? You know, day in, day out, our body encounters various kinds of foreign objects. It could be as small as pollen, it could be dust, it could be pollution, it could be many other kinds that we don't even know. So these changes in the antibody molecule helps to counter the side effects of those foreign particles in our body. So here the changes in nucleotides is mainly to generate flexibility. That's super flexibility. And try to watch these YouTube videos that I have. And then we move to how when a virus infects someone. Traditionally, every virus has its own life cycle, own plan of on or in other words, immune system had its own way to react to a virus or a bacteria. So when virus enters the body, they don't start multiplying right away. They try to sit and they try to figure out how best to capture the transcriptional and translational machinery of a host. right? And once but before, to figure out the transcription and translational machinery, the virus has to enter the cell, right? And then it has to enter the nucleus and enter the nucleus. So, so it takes its time to come into action, and that phase where virus adjusts, it's a very small phase where virus tries to, sense the immune system of that person, and the goal is to defeat the immune system, and it's trying to figure out its tactics. And then once it figure out the tactics, the main then that tactics could be to enter into the cell. After entering into the cell, cross the sea of cytoplasm, then knock on the door of nucleus and enter into the nucleus through nuclear pore, and then capture the RNA polymerase and translational machinery to produce active live virus. And then it is possible that immunity senses some foreign body and starts reacting. The first type of antibody that comes is IgM antibody, and then it's IgG. There are many kinds of antibodies, but here I would like to focus on IgG and IgM, mainly since it is mentioned on this slide. IgM is the earliest most antibody that is formed in our body, and it has a very short duration. It peaks and then it tries to, it generally reflects active infection, right? So let's say when we are going for RNA test for COVID or RT-PCR test, that RT-PCR detects the live RNA of COVID-19 that is in our nostrils, epithelial cells. So a positive RT-PCR test is a testimony for active infection, active ongoing infection in that individual. Similarly, at antibody level, the presence of IgM antibodies reflects active infection, not only for COVID, but many viruses. Because here, the immunity and the virus, the encounter between immunity and the growing virus is begun to occur, and IgM is that responsible for active, immune system reflecting active infection. Then IgG develops. IgG generally has a robust response more than IgM, and it stays longer. Now, why a disease establishes in our body is because these antibodies, they're not effective, right? They're not effective. There can be IgM antibodies, IgG antibodies, list of antibodies, but if they're not effective, then a person is going to have a disease, right? So this is just says that this development of cells, look at this slide in context of stem cells, that a bone marrow cells that is destined to become pro B cells, then pre B cells, then small pre B cells, and then ultimately they start secreting antibody once they encounter the pathogen. And once the pathogen has gone, the memory is still there. So we see the pathogen, our immune system sees the pathogen once and it has a memory, right? So that's the only part of immune system that helps to develop that is helping to develop various kinds of vaccines and immunotherapies against virus, bacteria, and also cancer. Right? So a particular foreign particle in our body is referred as antigen. But if you go into detail, a foreign substance is antigen when it can throw antibody. There are a lot of substance particles which are very small. They don't qualify the definition of antigen, but they can still create problem in our system, in our physiology. So a typical antigen could bind to or could be bounded by various antibodies. An antigen has an exposed surface. On this surface, various antibodies can bind. So in this pathogen or this antigen, there are 7 different kinds of antibodies here, 7 different kinds of antibodies. And the portion of antigen that reacts to antibody is called as antigenic determinant. Antigenic determinant. The portion of right. Okay. And then what happens that the T cells and the B cells, they collaborate with each other to make the immune system. So there are battery of cells in our blood that work together. B cells by antibody, T cells by cytokines, interleukins, neutrophils by porins, and then there are eosinophils, basophils, many other cells there involved in generating immune system or immune response. Cancer cells have been shown to, we talked about this in the stem cell, have been shown to express some unique antigens. And those antigens can be used, those specific antigens on the cancer cells can be used to target the cancer cells with specific antibodies. And these antibodies can serve as a starting point to react with cancer cells so that they don't grow, they don't divide. And then there are 2 kinds of antibodies, polyclonal and monoclonal. They both are good, depends what is the need, right? So monoclonal antibody could be good for like treating or for the killing of cancer cells, because monoclonal antibody will bind to only one site. So a cancer antigen which is specific to a cancer and an antigen which is specific to a cancer, they will bind at a specific point and start the cell lysis or cell death process. So, but polyclonal antibody could bind to various places on the cancer cells, or they may not be good. But polyclonal antibody could be good for diagnosis. So monoclonal antibody, so I'm talking in a limited. Human diseases, human body is very complex. One hat may not fit all. But talking in general terminology, monoclonal antibody could be good for therapeutics. Polyclonal antibody could be good for diagnosis. And also, price-wise, monoclonal antibody needs expertise, whereas polyclonal antibody, you can take a rabbit, a bunny, a rat, or a sheep, and you can just inject your protein, and then you can get antibodies. So, but monoclonal antibody, you need very aseptic conditions. You need 2 different cells to fuse. So you need to fuse B cells. B cells are present in spleen cells. In a spleen, so you get the B cells and you fuse with multiple myeloma cells. These are cancer cells. Myeloma is a cancer of B cells, plasma cells, which is the end stage of B cell development. So when you fuse, you get cells which can make antibody, monoclonal antibody, forever. They're immortal cells. They will never die because of the nature of its oncogenic activation. These myeloma cells, they have cancer gene activated that make them immortal. So that immortalness of this myeloma cell is being utilized to develop monoclonal antibody technology. And monoclonal antibody could be used for therapy, diagnosis, anything. One of the example is Herceptin, that is used to treat HER2 positive breast cancer cells. It's pretty effective. Patients get treated if they're HER2 positive. Cancer is a heterogeneous disease. And even within one person, there can be different kinds of cells. So it depends at what stage a cancer is diagnosed. And depending upon the stage, the heterogeneity of cells, the treatment process is planned by the oncologist. Antibodies, because antibodies are very specific. So if you develop an body against a cancer antigen, it will only bind to that antigen. It will not bind to anything else. So you can take the advantage of the specificity of antibody and make a chimeric molecule. So the V portion of the antibody can bind to the cancer antigen, and the FC portion here you tag a cytotoxic drug. Cytotoxic drug is those cells that can destroy cancer cells. So this groove portion, the V portion of antibody, will be specific to the cancer antigen. So it will target the cancer cells. And this cytotoxic drug will then enter into the cell and destroy. So it's very specific. Immunotherapy is the upcoming way to treat cancer cells. Now I'm switching gear towards diagnostics. Right now I was teaching about therapy in general, and especially reviewing what we taught last week. Here we are talking about diagnostics. So let me go and just give some basic introduction. So here we are looking at the diagnostic potential of antibodies. Diagnosis means to find out the cause of illness. Now, antibodies could be very helpful in knowing if there is an infection. Let's say if we want to find out the stage of cancer, so let's say whether a person is HER2 positive, or let's say in prostate cancer, what stage of development of cancer it is. It could be early, mid, late. So in different phases of the cells express different proteins. So antibody can figure out stage of cancer. And then stage of cancer is important for planning the treatment. So infectious agents, agents and stage of treatment. So let's get into the infectious agents, right? So how could the antibody be useful, right? So let's say if we want to, for sake of understanding, right? So let's say if we want to see whether COVID-19 is in our blood cells or not, right? COVID-19 is in our blood cells or not. So blood has a component which is mainly fluid, no cells, which is called as plasma. Right? So hold this thought, blood, the liquid, and here when you're collecting blood, you collect it in the presence of a chemical that does not let the blood coagulate. Right? When we draw the blood, put it in a test tube, the blood clots, right? If EDTA is there or heparin is there, then the blood doesn't clot. The other situation could be that we draw the blood, and then in the test tube, blood is in the test tube, or let's say no EDTA, blood is in the test tube. Then blood gets clotted and it releases yellowish fluid that is called as serum. Plasma is a pinkish color, or a pinkish colors fluid that comes out when you're having a blood in the plasma in the EDTA. If there is no EDTA, then it's serum. Now in the serum, serum is a protein, serum is actually a protein which is very rich in albumin. And this serum also contains antibodies. So if I want to know that that uh, that I am healthy, I did not get COVID infection or symptoms, so I feel that I never had, but I want to check whether I had or not. So if I had and I was non-symptomatic, I was healthy, means that how to check that? I cannot detect the RNA because I don't have infection, active infection. And even if I had infection, I never came to know about it because I was healthy. So the only way we can know is to check whether we have antibody to COVID or not. Right? Or let's say, if I had antibody, if I had infection, then what is the level of antibodies I have, right? If I have good amount of antibodies, then maybe I'll be protected next time, right? And especially we have vaccines. So vaccine is there inside our body and our body has reacted to vaccines. So we should have antibody to spike protein because mRNA is to is against designed against. So mRNA vaccine in case of COVID is designed against spike protein, right? So we want to check whether I have antibodies to COVID-19. Why I'm doing? Just see that whether I was infected in the past or not. If yes, what is the level of my antibodies? Is it strong? It is low, or the antibodies disappear? So what's going to happen in this situation is that we will need to take the help of a lab. So a lab, an immunology lab or a cell biology lab, a lab that has a potential, or a virology lab to grow COVID, that has a potential to grow, that has the facility to grow COVID-19 culture, right? And then one can take the COVID-19 in the tube, and then we can purify spike protein. Because if I have antibody to any part of COVID-19, it will not be helpful. I want to check whether the antibodies present in my body is against spike protein. So what will be done here is that we will take a ELISA plate. Right? An ELISA plate, there are 96 wells, something like this. And let's say a COVID virus or a spike protein looks like this. So this proteins will be adhered to this to the wells of the ELISA plate. This ELISA plate has COVID-19 spike protein. Now what you do, you take my serum that you got from my blood and put it on this in this this well of ELISA plate, and let it sit for maybe, let's say, 90 minutes to give the antibody to bind. Now let's say I have antibody. Let's say I have antibody. So this antibody I have is against the spike protein. So this antibody will bind to this spike protein, right? So what happened here? They formed antigen antibody complex. Now the challenge comes how to detect this antigen antibody complex, right? So we need help. And who helps here? Again, antibody. So we take the antibody, right? So an antibody is basically V-shaped, right? This is a heavy chain. Okay, and this is light chain. And this is the groove of the antibody to which an antigen can bind, this V-shaped. Now, what scientists have done, they have labeled this antibody with an enzyme. They have labeled this antibody with an enzyme. They have labeled this antibody with an enzyme. This enzyme is called as horseradish peroxidase. So this is now what happened? This particular antibody became a chimeric molecule. Why I say chimeric molecule? On one hand, it has antibody property to bind to antigen, and on the other hand, it has been made chimera by conjugating it to an enzyme called as horseradish peroxidase. Now, once this antigen-antibody reaction has gone for 90 minutes, you can bring this antibody here. Now, this antibody can only bind to the Fc portion of the this antibody. So our antibody, my antibodies will be something like this. This is the Fc portion, and this is the variable region, and this is the light chain, small. So this is my antibody. Now this secondary antibody will only bind to this Fc portion here. So there will be a circuit here. So let's look at the circuit here. So if you look at this slide, this is the antigen, let's say COVID-19 spike protein. This is my antibody, that is primary antibody. The secondary antibody can bind to the Fc portion of my antibody, and it is a chimeric molecule and which has an enzyme. So what does enzyme do? Enzymes, they can act on a substrate and can make a product, right? That's the definition of enzyme. So when this circuit is complete, right? If I have antibody to COVID-19, to spike protein, then the well of ELISA plate will show color. Where does this color comes from? This color comes when the HRP antibody in the secondary antibody reacts. So there is a circuit here. So let me just go over the circuit here. So this is an antigen. This is the antibody, and this antibody is bound by the secondary antibody that has the enzyme HRP. We add the substrate here. The substrate is colorless, but if the circuit is complete, meaning I have antibody. If I don't have antibody, then this secondary antibody will bind to nothing. So the reaction will not take place. If I have antibody, then this becomes the primary antibody. This becomes the secondary antibody. When we add substrate, then there is formation. So try to understand this plate. So let's take, so this is the brown color, right? Or yellowish, dark yellowish color. This yellowish dark color occurs because here enzyme and substrate, they reacted to make a product. So what was the enzyme? Enzyme was HRP. What was the substrate? It is called as DAB. And then when the enzyme acts on this substrate, then this becomes brown color or dark yellowish color, meaning that I had antibody and I was infected. Through this ELISA, one can also, many of us, we got vaccines, right? First dose, second dose, booster. Do we know whether we have antibodies to COVID-19? Do you know that how long the potential of vaccine is? Will the vaccine be effective after 2 years? Do we have enough memory cells within our body that when COVID again occurs, then we are protected? We don't know such answers. So this simple test can be done in across the population. Let's say somewhere there must be a data that how many people got infected in Brooklyn or Crown Street or Crown Heights. How many got positive? How many got vaccinated? They have the government, the healthcare, they all have the data. And what is the status of the antibody within the population? Can we compare the Brooklyn population to Queens population, Queens to Manhattan, from US to Africa, Africa to Asia? Can we do this? So these tests could be very helpful to understand whether there is any longevity to the vaccines. And it's a very simple experiment. You take the blood from individual, isolate the serum has antibody, and you make this test. So here in this well, one of the wells, you have the COVID spike protein, then you overlay the patient serum that has antibody, then you add secondary antibody, which has HRP. If this circuit is complete, then this color formation will take place. If the circuit is incomplete, then there will be no color. No color means no antibody. So, so these are simple tests, ELISA. Another test, diagnostic test that is done is Western blot, right? And Western blot so this is before I go to Western blot, this is the plate reader. It reads plate in few seconds. You keep this plate on this thing, you press enter, this will go in, and just like a printer, you'll get the results, and you can match the printout with the wells of the ELISA plate. It's very simple and straightforward. Right? Now, another technique that is used for molecular diagnosis is Western blot. Western blot is a technique where you use two different things. Number one is SDS page, polyacrylamide gel electrophoresis. This is different than agarose gel. Agarose gel is very, very less sensitive, right? And very simple. But here in polyacrylamide gel electrophoresis, what happens, it is more sensitive because you use very special in gel, which is called as polyacrylamide. And then what we do is that we make the gel here. Agarose gel, they run horizontally. They run horizontally. But the polyacrylamide gel, they run vertical. So you take, it's very tricky, and it's very, you need to develop the skill. With the practice it comes. You take two plates, in between the plates you put a spacer. You don't need to know these details. I'm just telling you. You take two glass plates, and you put a spacer, then you pour the gel, and then you run A vertical gel as compared to agarose gel, you add your protein samples in every well and then run the gel. The proteins will separate on the basis of their molecular weight. Now you can question me why not on the basis of charge? Because different proteins will have different charged amino acid. So the answer to this is that this system is called as SDS page. SDS page polyacrylamide gel electrophoresis. So let's say what happens here, that in polyacrylamide gel, what happens that let's say you take a protein, right? A protein has secondary structures. It could be alpha helix, beta sheet mixture. Now, this protein will have a mosaic of a positive charge, a mosaic of negative charge amino acid, or neutral amino acid. All the charges depending upon the nature of the protein. So this protein is then incubated with SDS. SDS is a detergent, sodium decyl sulfate. Now what this because of sulfate, this particular chemical has negative charge, and once negative charge, so once you add this detergent with the protein, so this negative charge will bind to the positive charge on the protein. So what happened? Protein does not have any positive charge because all this positive charge on the protein has been neutralized by this detergent. So because all the positive charge on the protein has been neutralized by this detergent, only thing left on the protein is negative charge. So the whole protein becomes negatively charged. So when you, in SDS page, when you do the gel electrophoresis here, all the proteins are negatively charged, so equal charge. In DNA is negatively charged universally because of phosphate moiety, but in protein, that's not the case, because out of 20 amino acids, there are some positive, there are some negatives, there are some neutrals, there are some hydrophobic, so you need to make them whole protein negative, and that happens because of SDS, the chemical. And then the protein is separated on the basis of molecular weight, the higher weight on the top, the lower weight in the bottom. And this is the spectrum of SDS page. SDS page is a polyacrylamide thin gel. It's very sophisticated. One has to be, it's sometimes, the right proportion of reagents and everything is very important. So once you have your protein on this gel, the protein is transferred on a membrane. And then this membrane is treated with antibody. So Western blot is a step higher than COVID. I repeat, Western blot is more specific than ELISA. In ELISA, it's quick. You can screen 96 patients because there are 96 wells. Western blot is more sophisticated. If you want to really do research to see against which protein a person is having antibody, whether it is spike protein or not spike protein. So let's say you want to see in the population of Brooklyn, or let's say among students of Medgar Evers College, how many of us we have antibodies to spike in staff, faculty, everyone? How many of us we have antibodies to spike protein? So the hypothesis of this experiment would be, do we have antibodies to COVID? If there is no antibody to spike, do we have antibodies to another protein of COVID? So when you have this series of questions, then you we'll do a study, we'll take out the blood, draw the blood, let the serum be, get the serum from each individual, do the ELISA, and then we find whether somebody is spike positive or negative. If somebody is spike negative, that doesn't mean that he may not have antibodies to another protein of COVID. So to find out that, you do Western blot. It's a little bit more, research oriented, more specific tool than ELISA. So we talked about molecular diagnostic, we talked about ELISA, we talked about Western blot. We have spoken in earlier classes, we have spoken of PCR, RT-PCR, right? So these are the molecular diagnostic, tools to establish whether the cause of the disease. If the cause is known, then the treatment is straightforward, right? And so, we talked about the role of monoclonal antibody in blocking COVID-19 infection, because these monoclonal antibodies, they will intercept the viral entry. So this monoclonal antibody to spike protein will bind to the spike and not let spike protein bind to the cellular receptor, and hence block the infection. Okay. So with this, we finish this chapter, and we are around 5:42. So I was thinking to start gene therapy, but let's finish this class early. I don't want to overwhelm you, but we can take this time to address any concern, questions, anything you want to ask me. So if there are no questions, then can I end this class?
Yes, Professor, and happy Ramadan.
Okay, same to you.
Have a good one.
You too. Thanks. Take care.
Professor, did you get me for attendance?
Name?
Anika Thomas.
Yes, I got you.
Okay.
What about me, Professor?
And me as well, Professor Bernard.
Yeah, Sagin Bernard. Okay.
Thank you.
Welcome.
Professor, can you?
Professor, did you get me?
Name?
Anderson.
Oh, okay. Yes, I got you.
Professor Rimshaw?
Who?
Rimshaw?
Yes, I got you, Rimsha.
Professor, can you clarify when the discussion board is due?
I don't remember. Can you check? It should be due pretty soon.
Okay, I don't think it says the actual date under the discussion board, that's why, but okay.
Yeah.
Yeah, Professor, I don't think the discussion board has like a due date to that.
Yes, that's.
Okay, I will check.
Okay.
But the assignment is due tonight though, right?
I don't remember the assignment's deadline. You have to check.
Okay. Yeah, the discussion board doesn't have anything next to it.
Okay. Okay, then if there are no questions, then I'm ending this class.
Oh,
Professor, did you get me on attendance?
What's your name?
Yael Nakhshomov.
Yes, I got you.
Thank you.
Professor, how about Aisha Morange?
Shamire. Yes, I got you.
Professor? Kayla again. I sent you last week when our assignment was due on Thursday night. I sent you my
May I know who I'm talking to?
Kayla.
Kayla, okay.
I sent you my assignment via email, but I didn't get a response back. I sent it before the deadline on Thursday
Yeah,
night.
so I mentioned before that I don't accept assignments via email. So you can submit with the next assignment and leave me a note on the Blackboard.
Okay, because I had Blackboard issues and I never had an issue before with submitting my
Yeah,
assignments.
so you have to contact the Blackboard because that's the best thing to do. And on the other hand, when your Blackboard is fixed, then you can upload with the next assignment.
So if the submission's gone, where should I upload it? In the same?
I said that with the next assignment, right? And just leave me a note.
Okay, so submit two in the same submission.
Exactly.
Okay, we'll
Okay.
do. Thank you.
Professor, I did the same. I emailed it to you, so I'll submit it both together.
Yeah, and leave me a note so I can see it.
Okay. We're allowed to submit twice? Like this, you can do multiple attempts under the submission?
No, I just told you that with the next submission. So
No, I know.
Let's say something is
just-
due on 12th. So you can put this together and you can write that you had a problem with your Blackboard and you're submitting here and you spoke to me during office hours. Or something
Oh,
like that.
submit it as one PDF. Okay, my
Yes.
bad. I get it. Okay. So I
One
believe
PDF, two
tonight.
PDFs doesn't, some students submit, in bits and pieces, some people to put together. So,
But one submission.
yes.
Okay. Okay, gotcha. No problem. Will do. Thank you.
Any other questions?